All right, thank you for the introduction. And I think those two initial talks really were helpful for laying out a lot of the things that I'm going to discuss today. I know many people in the room, but for those of you that don't know me, my background is actually in geology. I did my PhD in Earth and Planetary Sciences, where this is where I actually got my start in synchrotron using synchrotron XRF imaging, specifically of sulfur in ancient marine sediments to better understand the operation and evolution of sulfur cycling over Earth history. Nowadays, and during my postdoc, I actually do more environmental microbiome research. So I'm really interested in the cycling of phosphorus, sulfur, and potassium between minerals, microbes, and plants in a variety of different environments from terrestrial to marine settings. And importantly, one of the reasons that I came to learn about EMSL is I'm part of this collaborative outreach program at SLAC that is funded by the Department of Energy's Biological and Environmental Research Division, where it is my role to help new or novice users access some of our capabilities at SSRL. And so I don't think many people need more introduction on the importance of multimodal imaging. So I'm just going to go over this really briefly. But we all know that there's no one single tool that can answer all of the questions that we are interested in about a given system. And that's especially true for complex biological and environmental samples. So by combining tools, we can overcome the limitations of the independent techniques. And for Earth and environmental systems that we study, they're complex, heterogeneous, three-dimensional, and they evolve over time. And so this really necessitates having multiple tools for us to be able to visualize the key processes that we are interested in. And so there are several problems or challenges that Tamas laid out with multimodal imaging. And so I'm going to kind of talk very briefly about the couple of different types of multimodal imaging. Correlative, which is most of what we've seen so far. Most of what I'm going to talk about is correlative imaging. That is sequential imaging on a sample where image alignment and data alignment is required. And then there's simultaneous measurements. Simultaneous being you can actually take these images at the same time. And the main problem with simultaneous measurements is that we can't necessarily include a new type of imaging modality onto an already difficult imaging technique. And so with correlative imaging, there are several additional problems that we have to think about. And so these problems include the techniques themselves having different spatial resolution. The techniques could be destructive or alter your surface chemistry. And the data themselves could be really different, have different signals, and so could be hard to co-register. With that, and something that Arunima mentioned is that the sample preparation or the sample delivery to these different types of imaging techniques could be very different. And so we really need to think about having some type of sample platform and or sample holder that is somewhat universal. And so, of course, we potentially have some solutions. Arunima just nicely introduced the Terraform platforms, which I'm going to talk about some of my research using these, specifically the mineral micromodel and the rhizochip. And then we're going to hear from Sam about this, his software that he develops, the microanalysis toolkit, and we'll have a really nice demonstration from Sam tomorrow about how we can use this type of software to integrate our different imaging modalities. So I wanted to actually begin here with talking about chemical imaging using X-ray fluorescence microscopy at a synchrotron. I wanted to make sure that we were all on the same page with understanding how X-rays interact with our sample. So here I have my incident X-rays in yellow. When they interact with your sample, they can be scattered, they can be transmitted, and they can be absorbed. And it's actually this absorption process that we are looking at when we're doing X-ray fluorescence imaging. What happens is that you have an X-ray with a particular energy. That energy might be enough to excite an electron, particularly from the core level. This is very unstable to have a hole in the core level, and an electron will drop down to fill that hole, resulting in fluorescence. And this is what we're measuring when we're doing X-ray fluorescence imaging at a synchrotron. What I want to say here is this is very element specific. So that incident X-ray energy required to excite your electron is different from every element. And so this is how we can be really specific and probe specific elements that you are interested in your sample using this technique. And so I want to just very briefly go over how this type of imaging differs from something like a desktop XRF or SEM, EDX imaging. So this is a schematic of one of our imaging beamlines at SSRL. And I'm not going to go over just a few things on this diagram here. So in this in-hutch area that I have labeled, this might be where a user might load their sample and actually interact with. We have these axially symmetric focusing optics. At some of our microimaging beamlines, this means we can have a spot size as small as 1 micron. But ultimately, these help improve signal-to-noise ratio on a small spot on your sample. What's really important here is that we tend to have ambient sample environments. And so that means that we are amenable to different things like forming in situ reactions. And ultimately, we have relatively minimal sample requirements in terms of preparation, but I will mention those in a couple of slides. And so this differs to some techniques where in some imaging techniques, you require a vacuum or you require your samples to be coated for them to be conductive. I just want to point out one part of this upstream section, and this is the monochromator. The monochromator is what selects for that incident X-ray energy that I mentioned on the previous slide. This is what allows us to do X-ray absorption spectroscopy as well as X-ray fluorescence imaging at these beamlines, which permits the chemical imaging that I am interested in. I want to say that we actually have 3 particular beamlines like this at SSRL. They all have ever so slightly small differences from one another, being able to access a range of X-ray energies, as well as a variety of different spot sizes. And while I say that we have this ambient sample environment, we are able to accommodate different things like having an anaerobic chamber or cryo conditions. And so for those of you that have never done this type of XRF imaging before, this is just a little diagram of showing you how this actually works. In yellow here, again, is the incident X-rays. And then we can see that our sample moves over a particular area that you have predefined. And then we have various colors that are coming off of the sample. These represent fluorescence from different elements, and we have energy dispersive detectors that can tell us which elements that fluorescence is coming from. And so the result might be something that looks like this. This is a total sulfur map from a moss, an individual moss leaf. And whenever I show these maps, they're like heat maps or abundance maps, where the warmer, brighter colors are higher abundance and the cooler, darker colors are lower abundance. And so this is great, but you might be wondering how this actually is different to a different type of elemental imaging technique. And so what we can do after this type of imaging is we can go to an individual spot on our sample. Instead of moving the sample, we keep the X-ray beam fixed on that spot, and we actually change the X-ray energy from just below to just above the absorption edge of the element of interest. And the result is something that looks like this. This is a Zane spectrum, a sulfur Zane spectrum, specifically from that one spot that I have circled on the map. I don't have time to go into all of the details about how we do this chemical imaging, but we typically call it multi-energy mapping. And then we go from something that looks like the total sulfur map on the top to being able to create abundances of different chemistries. So chemical speciation. In this case, I'm showing in red reduced sulfur, in green sulfonate, and in blue sulfate abundance. So a few sample considerations. There are some sample requirements, but not too many. This is an example of a pea plant that I actually just grew on my balcony. I dried it, and while it was drying, I flattened it between heavy books. Because ideally, we have relatively minimal sample topography. So your sample topography should ideally match your beam size. It doesn't have to, but you can end up with some artifacts. This is our large format imaging beamline, and we can see that we can accommodate very large samples or several small samples. And we can see here, my small samples are mounted onto these small black inserts. These sample inserts are compatible with our microimaging beamline, so we can easily move samples from one beamline to another, and we can co-register those different data sets from our different beamlines. And so they're accommodated onto our microimaging beamlines by a multi-sample wheel. And these sample inserts come in a variety of different flavors, and we just 3D print them, and so we can accommodate various user samples. So what you can see from all of this is that your samples do have to remain upright and relatively stable upright for a few hours. Typically, we're working with solid samples for that reason. Most of the samples that you're seeing here don't have a covering or a coating. We can have some coverings if they are a particular X-ray transparent material to help your sample remain upright. And then importantly, if you have a matrix, so if you embed your sample in something like an epoxy, your epoxy really should not contain high abundances of the element that you are interested in. What's really important, and one relatively newer capability of our X-ray fluorescence imaging beamlines at SSRL, is being able to collect optical images of your samples mounted in the beamline. And so this is an optical image of a Brachipodium stem. And I'm showing this. I will also come back to this at the very end of my talk, and we might see this also featured in Sam's talk or his demonstration. But ultimately, this optical image is what helps us to be able to co-register and align data sets between our beamlines and with other imaging modalities. And so here, what I'm showing in red, green, and blue is the fluorescence of potassium and sulfur from our XRF imaging. Overlain in blue is that optical image. Okay, so now I'm going to get into some of the research that I've been doing with Arunima over the past several years. Potassium is a key nutrient to plants. It's associated with the movement of water, nutrients, and carbohydrates in plant tissue. It's involved in enzyme activation within the plant, and it's drought stress tolerance in a number of plants and microbes. However, potassium is often limited in the environment and in circumstances like the use of marginal lands or changes in nutrient bioavailability due to climate change. We need sustainable and cost-effective ways to increase potassium bioavailability. Yet, from this simplified diagram of the potassium cycle in the rhizosphere, we can see that there are several pools of potassium within the soil. And soils do contain up to 3 weight % potassium. The main issue here is that a majority of that potassium, up to 98% of that potassium, is considered non-bioavailable in this mineral structural potassium pool. So that is potassium that's found in minerals like potassium feldspars and biotite. And so while these do exist in soil, inorganic weathering of these minerals is too slow compared to crop growing seasons. One potential solution is that there are potassium solubilizing microbes. That means that they can actually weather these minerals in the structural potassium pool. And we know that there's a wide diversity of potassium solubilizing microbes, including many species of fungi and bacteria. However, of course, there are some problems in that the associations of these potassium solubilizing microbes and plants is not always positive. And of the available studies that are out there, there are quite a range of very different limited spatial and temporal resolutions. Additionally, we need to consider whether these potassium solubilizing microbes actually improve, whether the benefits of these potassium solubilizing microbes improve or decline in the presence of fertilizer. There have been some studies that shows that certain arbuscular mycorrhizal fungi, the benefits of them actually decrease when using fertilizer. And lastly, there are the molecular mechanism of potassium translocation, particularly within microbes, is not well understood. And part of the reason for this is that there is a lack of techniques that can directly probe and visualize potassium transformations and movements from minerals to microbes to plants. And so, of course, one potential solution that I'm going to talk about today is doing potassium XRF imaging and Zein spectroscopy. To further some of the complications that I already mentioned on the previous slide, soil heterogeneity and opacity is a central challenge in being able to visualize and resolve soil biogeochemical processes. And so like Arunima nicely introduced, EMSL researchers have been overcoming some of these limitations by developing and supporting the Terraforms platforms, which are microfluidic-based devices that simulate various aspects of soil physical and chemical properties. The two main types of the terraforms that I have worked with and I'm going to show some data from are the mineral doped micromodel and the rhizochip. And so again, we have been working on establishing these various workflows. Terraform's experiments, particularly the ones that I have been involved in, typically last up to 30 days, after which they can be partially deconstructed for different types of analyses. And so again, also like Arunima mentioned, these devices are sealed for the duration of their experiment, and they're sealed oftentimes by a PETE membrane. And so we can remove this membrane, and the membrane itself can actually be analyzed. We can then remove parts of the biomass. So if you have a plant, you can remove the above ground part of the plant. Then we'll have biomass that's actually left on the chip itself. And so with our XRF imaging, we actually realized that if we leave the biomass on the chip, that convolutes our potassium signal from the minerals that are in the platform and the biomass. And so we actually separate these. We do so by simply applying tape and a lot of pressure, and we're able to peel up and maintain the spatial relationships of our biomass that was in the chip. And so ultimately the result is that we have essentially 3 samples from any given Terraform's experiment. And so Arunima and I have been working together for several years to understand potassium uptake mechanisms from a mineral source by a saprotrophic fungi. And in these experiments, we started with a potassium-rich mineral-doped micromodel, like the one that I'm showing here in this diagram. And it was observed that fungi could only bridge between 2 nutrient ports, specifically when this potassium-rich mineral was present in the microfluidic device. And so this led us to ask various questions, such as what mechanisms did the fungi obtain the potassium? How is the potassium chelated for uptake? What form of potassium is transported in hyphae? And so to answer those questions, we turned to XRF imaging at SSRL and then MALDI mass spectrometry imaging at EMSL. And so we've kind of seen a few variations of this figure on some of Tamas and Arunima's slides. I'm just going to briefly walk you through this figure, where all the way on the left, I have total potassium. And we can see that I have these 2 individual spots on the lower part of the map. One is purple and one is green. Those correspond to 2 locations where I collected Zane spectra. We can see those in the middle. These Zane spectra themselves are different. And the bottom one, which is the purple dot, actually represents a potassium citrate. The one on the top, we're calling organic Zorb potassium or organic associated potassium. We believe that this is the main form of potassium that's transported in hyphae, and we think this is actually potassium that has various mixtures of different types of organic ligands and water. When we grew this same fungi on agar with no minerals and abundant potassium in media, we only saw that form of potassium, the organic Zorb potassium, and no other forms. And so here we can see that we have hotspots of potassium citrate that are specifically observed along the hyphae in fungi that have been grown on mineral-doped micromodels. And so since we observe potassium citrate and to a lesser extent potassium tartrate as hotspots, we suspect that this is associated with the uptake or storage of potassium. And so to understand how potassium may be sourced by fungal chemical processes, we mapped the spatial distribution of organic acids that were exuded in response to the presence of minerals on the micromodel. And so again, we've kind of seen this figure. This is a MALDI mass spectrometry image of 2 individual micromodel experiments, a 7-day experiment and a 30-day experiment. The main thing that I want you to take away here is that the spatial distribution of tartaric acid compared to malic, citric, and fumaric acids is different. And so actually what's happening is that the biosynthesis pathway of the fungal organic acids is actually dependent on the distance of the fungi to a carbon source. And so ultimately, we wanted to know what effect the organic acids have on the minerals that are embedded in our micromodel. And so to address this, we went back to SSRL and mapped the distribution of potassium-bearing minerals within the micromodel after 30 days of fungal growth. And so in this figure, and B and C represent the same area, and E and F also represent the same area, which is an area slightly further away from the inoculation point. In B and E, I'm showing the primary minerals that were embedded at the start of the experiment. So that is kaolinite, potassium feldspar, and biotite in red, green, and blue. And then in C and F, instead of biotite in blue, I'm actually showing you a clay, and that is a secondary clay that had to have formed throughout the experiment. And so these clays are primarily 10-micron coatings on the primary mineral surfaces. And so importantly here, the impact of the fungal oxidized organic acids has moved potassium from a non-bioavailable to a more readily bioavailable pool. And so this work until now has mainly been this correlative or sequential imaging on serial samples. That is, not everything I showed you was the exact same micromodel. But what we have been doing is really important and foundational groundwork, developing these workflows, testing what samples work with which modalities, et cetera. And additionally, this research until now has really been primarily focused on this individual saprotrophic fungi. And our goal is to increase the biological complexity of this system that we're studying using a rhizochip. We're going to intend to study the response of a bioenergy grass to drought with various microbial consortia. And so to best understand the rhizosphere processes controlling potassium cycling, we will focus on various multimodal imaging workflows of the rhizochips. And so this is the rhizochip here shown on the bottom, where we can grow plants within these chips that feature a soil porosity. And here's one of the rhizochips. It's kind of yellow in this image because of the minerals that we added. This is a mineral-doped rhizochip so that we could study how the impact of the plant roots interacted with the minerals. But in this data that I'm about to show you, I'm actually going to focus on the Brachydipodium stem itself, because we wanted to understand the transport of potassium between the root and the shoot of the plant. In this tricolor map, I'm showing you this organic sorbed potassium that we see again here in the Brachydipodium, as well as two other phases, a potassium phosphate and potassium nitrate. And I want to focus on the potassium nitrate in particular. We see this kind of purplish pink color. That's actually showing that potassium nitrate is co-locating with organic associated potassium on the very edges of the stem, as well as within hotspots of these hyphae that are grown on the stem. And so we're really interested in potentially the co-transport of nitrogen and potassium in this system. And so to better understand the local bonding environment of the organic compounds in the Brachydipodium, we turn to synchrotron FTIR at the ALS. And so synchrotrons themselves emit a broad spectrum of light, not just X-rays, and that includes the infrared region. The synchrotron IR beamlines at ALS have several advantages over a traditional benchtop FTIR. The primary one for me being that they are very similar in their resolution. So hopefully decreasing the multimodal imaging issues of resolution. And so I'm actually not going to show you data that are overlaid. I think Sam might show that either in his talk later today or tomorrow. But I just wanted to show you some of the images that we did collect on the exact same sample. They look a tiny bit different, but that's because in order to take the sample off of the IR holder, I had to use a small amount of water, and that actually impacted on the distribution of these hyphae. But what I was able to do is I was able to take the data from the IR beamline at the ALS and import that into SMAC and process it in a variety of different ways. And what I really want to highlight here is that these nitrogen-hydrogen bonds are really localized on that outer edge of the Brachydipodium stem, which is what I showed you, similar to what I showed you on the previous slide. And so this is just another way of showing various distributions from the IR data showing cellulose, protein, and lignin in red, green, and blue. And then this is the same image I showed at the start of the overlay of our optical image from our XRF beamline with the fluorescence of potassium and sulfur. And so I think, the main thing I want to say about our future directions here is really echoing some of the things that Tamas touched on. But those of us that do work at these national labs that are user facilities, it's really important for us to get together in situations like this, on various other workshops, where we can work on developing these workflows and pipelines to bring to the user community so that we're working on these. And then we also want to hear from the user community and how the pipelines they specifically need and how we can work to better accommodate those types of experiments. And so with that, I want to thank a large number of people, specifically the XRF imaging group at SSRL, a huge number of people at EMSL that I'm sure I'm missing some, as well as Neslihan, who's here from LBNL. She helped on the IR stuff. So if you have more questions about that, I will defer to her. And yeah, I'm really excited to be able to continue this work. We recently got an EMSL LSR approved to continue working on our drought studies.